transcription happen? Make a little more space. Does transcription happen? Okay. So, if we're looking at an inducible operon, we're going to have two possibilities. We could have no lactose. If there is no lactose in the media, in the organism, where is the repressor at? Is it on the operator or not?
It's on the operator.
It is on the operator. So it is bound to the operator. If it is bound to the operator, does transcription happen? No. Okay. It's physically blocking RNA polymerase from continuing. Now, if there is lactose present, where is the repressor? And really just, is it bound to the operator?
It's not bound to the operator.
It is not bound. It's generally just floating around in the cytoplasm. What is bound to the repressor?
Lactose.
Yeah. And it's really something called allolactose, but we're just going to say it. Lactose is bound to the repressor. And does transcription happen here?
Yes.
Yes, it does. So try to figure out where all the parts are. Okay. The other one we looked at was a repressible operon. And there we use that tryptophan operon, or the trip operon. And this operon makes tryptophan. Makes the enzymes that make tryptophan, but so you can think of it the same way. If there is no tryptophan in the cell, where is the repressor? The cell has no tryptophan. Does it want tryptophan? Probably. That's how it's going to make proteins. It needs that amino acid. So if there's no tryptophan in the cell, do we expect this to be transcribed?
Yes, because we need it, right?
That's right. Exactly. We need it. So we want to transcribe it. So do we think the repressor is bound to the operator or not?
No.
No. So it is not bound. So if it's not bound, does transcription happen?
Yes.
Yes. Right? RNA polymerase can make through. If the cell has a nice backlog of tryptophan stored up, where's the repressor going to be?
Bound to operator.
He's bound to the operator. And it does that because tryptophan is bound to that repressor. It's what's known as a co-repressor. Oh, bless you. Tryptophan is bound. And if this repressor is bound to the operator, does transcription happen? No. This is how I want you to be able to think about it. Now with lactose, there was a whole nother level of difficulty. Right? Where we talked at the very end of chapter 7 about catabolite repression. And what happens with catabolite repression?
Is that when, if glucose is present, other sugars are inhibited?
Right. So, and the way we usually think about this, if glucose levels are high and lactose levels are low, the lac operon is not transcribed, which that makes sense. there's very little lactose in the cell. Right? You want lactose. And you could do the same if glucose levels are low and lactose levels are low. You're not gonna have transcription. That still matches over here. Where this gets weird is if glucose levels are high and lactose levels are high, you do get a little bit of transcription, so it's not a full off, but it's a very low level.
So like, why you will get more of a stomach if you eat like ice cream?
That's different, it's just us being able to break the lactose down.
Okay, I was just like curious how this works when you're like, eating something that has a lot of sugar, but it also, like a lot of glucose, but it also contains lactose. Is that when it's gonna just make a little bit of lactase?
So this is more for E. coli.
Okay.
We do this completely different.
I was just, okay, I'm curious how this is applicable to people, but,
Yeah, we actually break down lactose in a whole different manner,
okay.
so, I mean, you know,
Thank
you're
you for
gonna-
the clarification.
Yes,
So it's completely different?
it's a completely different thing with
Alright.
people. The E. coli in your gut would have this going on if you had a lot of sugary ice cream. But us, yeah, we'll break it all down.
Okay.
Fourth possibility is when the glucose levels are low and the lactose levels are high. And that's really what we're describing over here, where you get a high level of transcription. And we're not gonna go into the molecular behavior behind that. We could, but we're not going to. We did that 161 actually,
Yeah,
which was
I do
insane.
remember that a little bit, yeah, as we're going over this.
That was insane. All right. Any questions on this? This is trigger memories from Monday, hopefully. Definitely will be a couple questions on your next exam about this. The last thing, and we didn't talk about this for a while, but I can just use the blue one. So, RNA polymerase binds, nothing on the operator, it goes through. Remember, it's gonna make one long RNA. 5', 3'. This RNA will have every single gene on it. So this is the weirdo thing bacteria do. A, B, C, I don't know. We'll give this one 4 genes. It's what we call a polycistronic RNA. And again, bacteria and viruses only. And then, when it gets time to do translation, that's where you'll get the different proteins here. Whatever, we can make all sorts of different proteins, which will give the 4 different proteins at that point. If it's a virus, viruses will sometimes make one long polypeptide and then cleave that to make the different proteins. So, one of the big anti-HIV drugs is a protease inhibitor that actually stops it from cutting up its long polypeptide. That's enough bacterial genetics. Questions on all of that? Okay, so what I'm going to do, now that I draw all this, is I'm gonna erase it again, because otherwise we'd have to move to this other board. So take a second, if you need it. And we're gonna look again, let's see, we started with mutations on Monday. DNA had 2 types of mutations that we talked about. What were the 2 types of mutations in DNA?
Horizontal gene transfer.
So before that, we have a point mutation, which if you've learned mutations in the past, you might have learned as a base substitution. Is another name for it. And that is where one nucleotide is replaced with another. So like an A is replaced with a G or something. What was the other one?
Synonymous mutation?
So that's gonna be one of our protein results from this.
Is it the nonsense?
That's another one of our protein results. I don't even know.
Oh, spontaneous? Is that what you're looking for?
Frame shift mutation? We have a lot of mutations listed here.
I know.
So,
So what causes a frame shift mutation?
it says it's usually result in nonsense.
Before
Not-
that,
Like,
what causes it? What causes the frame to shift?
Addition or deletion of one or two base pairs?
Yeah. So, DNA mutations really come in two flavors. You can change a nucleotide, or you can add or lose nucleotides. Otherwise, you have the bigger one where you break the backbone, but that's a bigger problem. Right. Point mutations, gonna give you one possibility is a synonymous mutation. What's a synonymous mutation?
Nothing changes.
Right, nothing changes, the amino acid sequence stays the same. And so there's really no effect on the cell. What's another possibility here? We have three possibilities.
Missense?
A missense, and what happens in a missense? So if a synonymous or silent mutation is nothing changes, what would have happened in a missense?
says a different amino acid is inserted.
Yeah, you get a different amino acid. And this can have a range of effects. It can have almost no effect. Usually it's detrimental. Usually the protein either completely loses function or partially loses function. But in the rare case, you can also gain function. Sometimes the protein gains a new function. Sometimes it gets better at what it does. All right. What else could a point mutation do? You get a nonsense mutation. And you're either gonna add or remove stop codons. And what does that do to the cell? Or to the protein? Would that be a good or bad thing?
It's bad.
That would be really bad. Messing with stop codons makes the protein either really short or really, really long, and it'll never fold upright. So these are very detrimental. Those are your three possibilities when you change a single nucleotide. When you add or delete nucleotides, what was that type of mutation called?
Frame shift?
That was the frame shift. Everything past that shift changes. Every single amino acid. That's usually extremely detrimental as well. Frame shift mutations tend to be carcinogens. Things that cause cancer tend to cause frame shift mutations.
Is that within the bacteria?
That's within everything. So even us. This applies all over. So I don't think we really do, unless you take the genetics class where they talk about different diseases that are caused, some by base substitutions, some by additions, deletions. We don't do much with that in our other courses here. That was the summary of what the changes are. Then we did do a little bit about what causes these changes, very briefly. We had spontaneous. What's a spontaneous mutation? What does that mean? So, it does just happen, but like, so when does this happen in the cell? I'm trying to think of ways to direct this. Is it like when your cell's just sitting dormant?
During normal divisions.
It's normally during DNA replication and cell division, where DNA polymerase makes a mistake. That's spontaneous. Then we had our chemical mutagens. And I don't care that you know any of the actual examples there. But some chemical mutagens are gonna cause base substitutions. Some are gonna cause additions or deletions. And then we had really one other major type of mutagen. What was our last type of mutagen? It's also a microbial control method. Radiation is your other mutagen. That's why it's a good microbial control method. UV light, what does that do to the DNA?
Causes thymine dimers.
Thymine dimers, right? Adjacent thymines on the same strand covalently bond. Usually that's gonna lead to an addition or deletion. What does ionizing radiation, like X-rays and gamma rays, cause?
Cancer.
They do. Why do they cause cancer? So they actually break the sugar phosphate backbone of your DNA. So you normally have how many chromosomes? 46. Start breaking those up so that there's a lot more in the cell, just random pieces. And that is extremely deadly to the cell and usually to the organism. Okay, how is that big summary? Questions on those? What was the last thing we did? We did one more thing. One more thing. Oh, I gotta look back.
We did the repair of damaged DNA. And we did horizontal gene
Oh,
transfer.
horizontal gene transfer, that was where we finished. Yeah. How bacteria change their DNA. Right, we talk about how humans, how do we have such varied offspring? We had two processes.
Right?
Not mitosis.
Meiosis.
Meiosis,
What
And
happens?
meiosis.
So what happens in meiosis? It causes so much variability. You know, Sarah knows, she told me on Monday.
Crossing over.
Crossing over, right? Prophase one, homologous chromosomes, exchange material. And then there's one other part, not really meiosis, but the end product of meiosis that can lead to variability in offspring. Why do you not look like your brothers and sisters? Random eggs and sperm come together and no two egg and sperm are the same. So that random fertilization. Bacteria are haploid. They reproduce asexually. So really, their sources of genetic change are mutation, and then that last thing we talked about, that horizontal gene transfer. Right, we had 3 types. What were the 3 types? And they're actually on the overhead if you don't remember. We put up little videos of each one in case you are like the videos.
Transformation, transduction, and conjugation.
Transformation, transduction, and conjugation. What happens in transformation? Yeah, so cells that are called competent, they just pick up DNA from the environment.
What do you mean by naked DNA?
It's literally just DNA in the environment. So it's not surrounded by protein or lipids or anything.
Is that within the cell or like outside of the cell?
It can be outside of the cell. So think of like things just in water. And like if they're in a big clump of bacteria in the water, if some of those cells lyse, the other cells around them can pick up the DNA from them. What has to happen for the cell to keep this DNA?
It has to be incorporated into the chromosome.
Yeah, the DNA, it has to be incorporated, and it could be the chromosome or plasmid incorporated into a chromosome or plasmid. Doesn't matter, but it can't hang out on its own. Can all bacteria do transformation naturally?
No, only a few.
Yeah, only a few can do this naturally. But in the lab, we can drive a lot of cells to do this. It is an extremely discouraging process to teach a bacteria to do this. That was one of my graduate school things. It was annoying. Transduction. What is transduction?
DNA transfer by phage.
Yeah. So DNA transfer by phage. And what has to happen for this to stay in the cell? Does it just stay on its own? It's usually linear. Linear DNA does not stay in bacteria. So this same thing usually has to happen when you have DNA transfer by phage. It has to be incorporated. Can all cells benefit from transduction? All bacterial cells? Yes, they can. In fact, we benefit from transduction from animal viruses. If you want to call it a benefit, sometimes we benefit. Yes, they all can. All right. Last one, conjugation. What was conjugation?
Two cells literally join.
And what does one give the other?
The F plus, right?
Yeah, the F plus cell gives what to an F minus cell? It's not, I mean, it is going to give DNA, but it's a specific form of DNA.
The plasmid?
It gives a plasmid. We actually call it an F plasmid or conjugative plasmid to an F minus cell. Once it gives that plasmid, then the receiving cell becomes an F plus cell. Since that's a plasmid, it does not have to be incorporated into anything. It's a self-regulating, self-replicating structure. It is stable as a plasmid. So no need to incorporate it into the chromosome. It's why these are so rare, because they need that incorporation step. And this is very common, because that plasmid's going to hang out. Can all cells participate in conjugation? No, they have to be able to go through this process where the two cells come together. So this is also a not all. It does seem like the more we look, we can find conjugative plasmids all over the place. But there's no guarantee. Okay. That was a quick summary of almost everything you've had so far. Questions on that? All right. The last bit for this next exam is a overview of immunology. If you've had anatomy physiology that covered immunology, this is going to be easy because they covered it in more detail than I probably am. The only things you'll learn different from me are ways microorganisms evade our immune system. But I'm going to start, just to keep it light a little bit, with a video. It's only 7 minutes. Hopefully my sound's working and not too loud.
This patient's cold is caused by rhinoviruses, shown here in red, that entered her nasal passages and escaped the first-line innate defenses, such as mucus and cilia. Some of these rhinoviruses entered the respiratory epithelial cells, where they replicated, releasing more rhinoviruses into the patient's nasal passages and tissue fluids. In the tissue fluids, cells of the second line of innate defense, such as this dendritic cell, carry out surveillance for anything that appears foreign. Dendritic cells engulf the rhinovirus within an endosome. The endosome fuses with a lysosome, whose enzymes digest the rhinovirus. The resulting rhinovirus fragments are called epitopes. The dendritic cell then attaches the rhinovirus epitopes to presentation molecules called MHC2. This complex folds outward so that it sits on the outside of the dendritic cell membrane. The dendritic cell then enters a lymphatic vessel and travels to a nearby lymph node. Here, the dendritic cell, part of the innate defenses, encounters a helper T cell, which is part of the adaptive defenses. The T cell receptor, or TCR, of this helper T cell recognizes the rhinovirus epitope, while the CD4 glycoprotein associated with the TCR recognizes the MHC2. The dendritic cell then stimulates activation of this helper T cell. Adaptive defenses help to control the current infection, but also provide immunity against future infections by the same rhinovirus. They are carried out by two types of lymphocytes, B cells and T cells. Immature B cells conduct surveillance in the lymph nodes, using B cell receptors, or BCR, to recognize only one specific epitope. The BCR on the left does not recognize the rhinovirus, but the BCR on the right does, and this B cell is selected for activation in a step called clonal selection. The selected B cell takes in the rhinovirus, degrades it, and presents the rhinovirus epitope with its MHC2. Our mature helper T cell recognizes the rhinovirus epitope with its TCR and the MHC2 with its CD4. Then the helper T cell secretes chemical messengers known as cytokines
Science.
that activate the B cell. The B cell proliferates and differentiates in a process called clonal expansion, resulting in two types of cell. Memory B cells carry the memory of this pathogen for many years. Plasma cells secrete antibodies that conduct surveillance for more rhinoviruses, grouping them together so they are easy targets for phagocytes. Antibodies in the blood and tissue fluids seek out free rhinoviruses. The antibodies neutralize the rhinovirus by binding to its surface molecules, thus preventing attachment to host cells. Antibodies are humoral defenses that target free pathogens. But what about the rhinoviruses that are already inside our patient's cells? Another type of adaptive defense cell, called a cytotoxic T cell, can locate and destroy rhinovirus-infected cells. It provides cell-mediated, or cellular, immunity. Let's return to our patient's lymph node to see how cytotoxic T cells become activated. The T cell receptor, or TCR, of this particular cytotoxic T cell is genetically programmed to recognize the rhinovirus epitope when presented by the dendritic cell. This time, the epitope is presented differently, with an MHC1, which is recognized by a CD8 glycoprotein on the cytotoxic T cell. With the help of cytokines from a nearby helper T cell, the cytotoxic T cell is activated and clonal expansion occurs, resulting in some long-lasting memory cells ready for a subsequent exposure, and many active cytotoxic T cells, or CTLs, that leave the patient's lymph vessel to search for infected cells that present rhinovirus epitope with their MHC1. The epitope is recognized by the TCR of the cytotoxic T cell, and the MHC1 is recognized by the T cell's CD8 glycoprotein. The cytotoxic T cell then secretes perforin molecules, which form channels in the infected cell's membrane. Granzyme, also from the T cell, passes through the perforin channels and activates enzymes that lead to apoptosis, the programmed death of the infected cell. The cytotoxic T cell then moves on to look for another infected cell. Together, the humoral and cellular defenses are the body's third line of defense in fighting a very specific pathogen. The humoral defenses, ultimately in the form of antibodies, conduct surveillance for rhinoviruses that are outside the body cells, in the patient's tissue spaces and bloodstream. The cellular defenses, in the form of cytotoxic T cells, search for rhinovirus-infected cells, inducing apoptosis when they are found. Memory T and B cells help our patient's body to recognize and more rapidly fight the same rhinovirus should it infect her again.
So, we're gonna get started on this today, and then we'll hit a lot of those details next week. Again, if you've had immunology and anatomy physiology, hopefully that's fairly review.
Brings up bad memories.
I'm so sorry.
I haven't taken that yet.
Well,
We're gonna-
you'll hate it just as much there as you do here.
As I say, they'll probably do it even in more detail than I do. I don't know.
They
Or-
just go over like leukocytes, so like all the stuff related to white blood cells and how it fights infection.
Yeah, so we'll do that on a skimming level.
Yeah,
Instead of on a deep dive level.
but I wish they would it would help if they put it all together, cause like this is, you're seeing a different side of it
They
here than you
didn't
do.
put it,
You don't get like the microbiology aspect of it, it's just like, what does this do?
oh, well, it doesn't make it as, I don't know. I think you'd have to put it into,
You
what is
just-
it attacking?
You go over, 'cause you learn about all of that when you're learning about blood.
Right, components,
Mhm.
okay. Well, we're gonna put it together, talk about how it works. We're gonna start with a lot of definitions and a lot of new parts, okay? If you are a note card person, flashcard person, this is the chapter for you. So, immunity is really what happens when our body gets off. It doesn't always have to be an infection, because you're in microbiology, we're gonna deal with it in terms of an infection, both viral and bacterial, and even a little bit parasitic. Parasites being like protozoans if you drink bad water, or parasitic worms if you eat generally undercooked meat. We're gonna break it into two parts, just like they did in the video. We're gonna have our innate immunity. Innate immunity is present at birth, and it will defend against anything. In fact, some of your innate immunity is there before birth. Right? But we just say present at birth. It's not specific. So it doesn't recognize specific pathogens, it just recognizes, hey, this doesn't belong here. It also often cleans up dead cells and other just random material. Right, so it's not specific, it's present at birth, and it's very rapid. This is what happens first when you get an infection or an injury. So we'll start on that today and we'll finish it next week. On the other side is our adaptive immunity. And adaptive immunity is why, as little kids get older, they get less and less sick. It's why we use vaccines. Adaptive immunity develops, usually during childhood. You can get some as an adult, but it tends to be at its max during childhood. And adaptive immunity is generally very specific. I remember in the video how those B cells and T cells recognized epitopes from the specific virus. So each cell is actually specific to something. So they recognize actual infections. And adaptive immunity is very slow. First time you are exposed to something, it's usually 7 to 14 days before this really gets going. Again, that's why we use vaccines, because future times you're exposed to it generally takes about a day to ramp it back up. Right, but it is very slow. Within the adaptive immunity, that's where you have antibodies and antigens or epitopes. So when you think about antibodies, you're thinking about here. I'm going to introduce the other word, epitopes. So an antigen is like maybe a flagella on a bacteria, right? Big, long structure. The epitope is the actual tiny piece of the flagella that an antibody binds to. So epitopes are extremely specific. Antigen's kind of a bigger term. Our adaptive immunity is also the part that will destroy our own cells, like if they're cancerous or they're infected. Remember the T cell in the video? So these will also destroy self, is how we put it. So either they're infected or if they're cancerous. So we do have a whole part of our immune system that deals with cancerous cells. We always think of the immune system and what's going on in us as biological warfare. It's your immune system fighting against pathogens. It's your immune system fighting against cancerous cells. And as long as your immune system stays ahead, you stay fairly healthy. And the image is what a dendritic cell actually looks like. If you were curious, they've got all those crazy arms, so it gives them lots of surface area. Right? Just like with all the organelles having all the twists for surface area, all these crazy arms are covered with those receptors. So lots of surface area to present things. I'll probably talk about these on Monday. Okay. Questions about these two parts? So this first chapter, I never remember what number it is, 14 is innate, and then chapter 15 will be the adaptive. And again, I'm going to give you so many crazy terms. I blame the immunologists for making this sound more complicated than I think it needs to be, but they love acronyms. Okay. Within this innate immunity, we usually say there are 3 lines of defense. If you read your textbook, it kind of does it like building security, which is kind of an interesting analogy. But the first line of defense is kind of like walls, right? It's going to keep things out. The second line of defense is kind of like a security camera, something that's out looking for infectious agents. And then your third line of defense is more like the actual security personnel. The cells that go out. I can't spell today. Security forces. I think they call them SWAT teams or something, but they go out and actually kill the invading organisms. Okay. So some of the terms here. Your first line, your skin. Okay. And we'll look at this more. Your mucous membranes. So most of your internal organs are lined with mucous membranes. If you think from your nose, clear down through your lungs, there's a membrane there, it's covered with a layer of mucus. From your mouth, clear to the other end, membrane's covered with mucus, right? Those are all gonna be the first line of defense. Not gonna let things in. In addition to these, our skin and our mucous membranes are covered with antimicrobial substances. Think saliva, think tears, think earwax. All of those are antimicrobial substances. We will briefly talk about them. We could do a whole class just on these substances. Right? Here's where I think the textbook makes it too hard. Security cameras, it calls sentinel cells. Things like that dendritic cell are actually a sentinel cell. They're out trying to identify, let me make sure I spell this right, pathogens. Right? And what sentinel cells are looking for are pattern recognition receptors, or they're using pattern recognition receptors to identify pathogens. Your book, if you read it, will give you lots of different pattern recognition receptors. I'm going to introduce you to one called a toll-like receptor, just because it's the most common. But use pattern recognition to identify organisms, or to identify microbes, or to identify pathogens, whatever. So these are going to say, hey, look, there's a pathogen out there. The other thing we're going to talk about with this second line of defense, really important, is called the complement system. So this is number one, this will be number two, be our complement system. Which when first discovered wasn't thought to be much of anything. We've realized what a huge role this system plays in maintaining our health. The complement system are just proteins made by the liver that basically circulate throughout the blood and lymph that look for pathogens. I was going to say proteins secreted by the liver for now. And it's found that a lot of people that have a less functional immune system is because one or more of these proteins, they don't make them. We've actually found that's a huge part in protecting us. All right. Questions on those? So we're going to hit each of these in detail next week. Right. Some of the processes that are used and chemicals that are used here. We're going to use, we'll talk about that third line later. Interferons are chemicals released by cells when there is a viral infection, at least most interferons. And I will come back to them when we talk about chemicals that are involved. But, So interferons, they're released by virally infected cells. Okay, I'm just trying to get some of this terminology out here. The other term I want to introduce from this overhead, just so when I talk about it later, it's not confusing, are what are known as phagocytes. Does anyone remember what the word phagocytosis means?
To eat.
Right, cell eating. So it's cells that eat other cells. Not many of our cells are actual phagocytes. All right, so we're going to introduce here as our security forces are phagocytes. Cells that eat. Usually they eat other cells, but they will eat all sorts of things. So these are gonna be our phagocytes over here. Okay. Questions upon kind of this nice introduction? Hopefully it's a nice introduction. We'll talk about inflammatory response and fever at the end. All right, you know what? I'm gonna stop there, and then on Monday, we'll break into the details of this. On Wednesday, we'll break into the details of the adaptive response if we get that far. Questions? All right. I will see everyone in, I'll get about 5 minutes, I'll get the lab opened up. Start actually with a discussion of scheduling.